All right, we're moving into the part of chapter 12 that talks about how neurons function. So we're going to talk about membrane potentials. These are the changes in charge that occur at the membrane of the neuron that allow them to send in a message very, very quickly. These include the resting potential and the action potential. We'll talk about how they happen, what's going on there, what molecules are moving, and what's happening at the cell membrane. You should be able to describe both the resting potential, which is shown here at minus 70 millivolts, and then the change to the action potential, which is the depolarization and repolarization of the cell. And we want to be able to talk about the sequence of events that cause this to spread across the surface and describe the movement of both sodium and potassium ions. And so here, again, a reminder of neuron structures. Neurons can receive and transmit electric messages across their surfaces. The dendrites provide the receptive surface. This is where the action potential starts, and then it spreads to the axon. Okay, these are electric signals. Okay, neurons, like muscle cells, have an imbalance of ions across their surface. These are membrane potentials. They generally have more sodium on the outside, which sodium is a positive ion, and more potassium on the inside, another positive ion. But since there's more sodium, the cell has a negative charge overall because there's more sodium on the outside. Generally, it's between minus 70 and minus 90 millivolts. And these ions are lined up right on the surface of the membrane. Okay. In the action potential, ion channels open so that sodium rapidly flows inside, and that's a depolarization because the cell membrane actually goes towards zero and actually a little bit past it. And this spreads because other ion channels open across the cell, and these are sodium channels, and they're only open briefly. And then they close and the cell repolarizes, and so eventually getting back to the resting potential. So I have to talk about what allows the cell to do this. And neuron membranes are regular cell membranes. They have primarily made up of phospholipid molecules arranged in a bilayer, but then they also have a bunch of proteins embedded in them. These proteins include several different types. One shown on the bottom here is referred to as the sodium-potassium exchange pump. Okay, and this moves 3 sodium ions out and 2 potassium ions in. So the sodium goes out and the potassium goes in. You can see that at the third one there. This does take some, it takes some energy in the form of ATP. And then the other thing that happens is that some of these potassium that move in leak out again through what are called leak channels, shown at the top. And these leak channels mean that, again, there's an excess positive on the outside and then negative ions on the inside. Okay, the other thing that these membranes have is they have what are called gated channels. And these channels can be opened or closed, and what causes them to open can differ. Gated ion channels, and there are gated ion channels for both sodium and potassium, are what's responsible for the action potential. So let's talk about those and show some pictures of those. First, these in the upper part, it shows what's called a ligand-gated ion channel. And these occur on the dendrites, and they allow the cell to receive messages from other cells. These open and close when a specific molecule binds to the surface of the ion channel. That molecule or message sender is shown in red in the upper photo or upper illustrations here, and it's called a ligand is a general term for that. And when that binds to this ion channel, it causes it to open. So it's closed in the upper left, and then the ligand binds the ion channel and that opens it in the right. And that allows molecules to move through. Okay. And so these are the types of ion channels that you find in a neuromuscular junction. You also find them in junctions between nerve cells. And then below, we have voltage-gated ion channels. These channels open when they sense nearby changes in charge. Okay, and so these channels are in nearby areas to the ligand-gated channels, and when ligand-gated channels open, that change in charge is sensed by sensors by the voltage-gated channels, and that causes those to open. And so here you see them on the lower left closed, and then on the lower right open. And so these are common in both nerve and muscle cells, and they allow the changes in charge to spread. And so when a ligand-gated channel opens at a dendrite, this will cause it to spread from different places. And so when an action potential happens, it occurs when there's enough change in charge at the membrane to pass a threshold. Okay, and that threshold is generally minus 55 millivolts. And when that happens, all the voltage-gated channels will open, but they only open in nearby areas, but that causes it to spread. Okay, and sodium channels open quickly, and so you get a quick depolarization that occurs, causing nearby channels to open. Those channels then cycle closed, and potassium channels open, and those channels allow potassium to move out, and that potassium channel will cause the repolarization, or here the 0.4, the 4 phase on the diagram. So the depolarization is caused by the opening of sodium channels in 2 and 3 of these illustrations. And then the repolarization is caused by the opening of the potassium channels. And those potassium channels remain open long enough for you to get a kind of a hyperpolarization or an afterpolarization. And so this is what causes the kind of quick change in charge and return to the resting potential, is that you have these two voltage-gated channels spread over the surface of the membrane. And so as the different parts of the membrane reach threshold, these things are opening and closing these gated channels. And this happens very quickly in milliseconds. And so action potential quickly spreads over the surface of the neuron. There are some special sequences in myelinated axons. In these cases, the myelin covers most of the axon, and there are just little gaps called nodes of Ranvier between the myelinated cells around the axon. This causes the action potential to jump from one node to another, which is much quicker than spreading evenly across the axon. And so here the ion channels are concentrated at these nodes of Ranvier. This is sometimes called saltatory conduction because the action potential jumps from one node of Ranvier to another. And so you see that very, very quick change in charge. And this is happening in motor neurons. It allows motor neurons to send impulses much, much faster than other cells. So in summary, neurons maintain electrical and chemical gradients by the movement of ion pumps. And so this sodium-potassium pump and the potassium leak channels cause the membrane to be at minus 70 millivolts at rest. Okay, gated ion channels for sodium open to depolarize the cell. This causes an action potential. The neurons quickly repolarize when the sodium channels close and the slightly slower potassium channels open. And so this causes an impulse to travel very quickly across the entire surface of the neuron. Next time, we're going to talk about synapses and neurotransmitters, and we'll have a lot to say about those. And that's the other big topic that we have to cover on how neurons function.